today is a little bit hot in Spain, but I hope in Bilbao it's a little bit cooler.
It's also hot.
Here at the office, I have 38 degrees, so it's okay. But anyhow, today we have the pleasure to have Jorge Simón. Jorge Simón is a postdoctoral researcher at the lab of liver diseases at ISCiii bioGUNE under the supervision of Malu Martinez-Chantar. So he has published a lot lately and in very high impact factor journals such as Cell Metabolism and Journal of Hepatology. He's very interested in the role of glutaminase 1 and cyclin M4 and how it affects the role in the progression of non-alcoholic steatohepatitis. And lately, he published a very nice paper on the role of MIR 873, right, Jorge? And acetaminophen. So he's going to give us a background on what he has done and the targets that he has identified as fundamental in the progression of DIli and liver disease as well. So Jorge, whenever you want, you can start.
Okay, I will share the screen. Can you see it?
Now we can see it perfectly.
Okay. So first of all, thank you, Javier, for the presentation and invitation. And as you have said, in this talk, I will give a brief overview about the effect of the microRNA, especially the 873, in drug-induced liver injury, and how a treatment based on this microRNA rewires the homeostasis of ammonia, and then it protects the liver from acetaminophen. First, I would like to give you a brief overview of about the modulation of ammonia in the liver, and also the role of the methionine cycle in the liver. Probably most of you already know them, but it's always good to remember. And then I will present the role of microRNAs in GNT modulation and in liver diseases. And then I will present the work. And I would like also to give some points of the role of the microRNA, not only in drug-induced liver injury, but also in hepatotoxicity derived from alcoholic damage. Ammonia, the liver needs to maintain an adequate ammonium balance in the organ. If not, many ammonium alterations have been related to liver pathologies and also to other related pathologies from the liver, such as hepatic encephalopathy. On one hand, we have the production of ammonia, mainly by the action of glutaminase. The glutaminase is placed in the periportal region and it's responsible of glutamine catabolism. Glutamate can enter to other pathways. It can enter the synthesis of glutathione or to the TCA cycle in order to fuel the cycle in an aprotic reaction. And on the other hand, it's important to scavenge the produced ammonia. For this, we have two pathways. One is the urea cycle. It's placed in the mitochondria and in the cytosol of periportal hepatocytes. And also in the perivenous region, glutamine synthase also has the capacity of scavenging the ammonia by conjugating it to glutamate in order to synthesize glutamine. Well, another pathway is the methionine cycle. It's essential for the correct functioning of the hepatocyte. And the methionine enters the cycle, or it is synthesized from betaine. Then methionine is metabolized by MAT1A, MAT2A, or MAT2B into SAMI, which is the main methyl donor in the liver. Then GNMT is the responsible of SAMI catabolism to S-adenosyl homocysteine, and then it enters in it can be recycled in order to further produce methionine. As you can see, this cycle interconnects with other pathways, such as the folates the sarcosine and the transsulfuration pathway. And here, homocysteine is of relevance because the CBS leads to cystathionine formation for further glutathione synthesis. As it is essential for the correct functioning of the hepatocytes, perturbation in this cycle are widely related during liver pathologies. In fact, with the animal models which are from MAT1A or GNMT spontaneously developed NAFLD, fibrosis, and even hepatocellular carcinoma. Related to the microRNAs in the modulation of methionine cycle, especially in GNMT, these microRNAs, several microRNAs have been related to liver diseases, and they are involved in many essential processes, such as inflammation, epithelial mesenchymal transition, or the activation of hepatic stellate cells in fibrosis. Our group hypothesizes that during liver pathologies, we have a GNMT downregulation. So, focusing on the relevance of microRNAs, a previous researcher from our lab hypothesized that probably microRNAs may be modulating the GNMT downregulation. And in silico analysis showed that this microRNA probably bound to the expression region of the gene. This was further demonstrated in this work. One of the first experiments performed is a luciferase assay, in which shows that when expressing the mimic microRNA, which is a microRNA with a similar function than to the microRNA, there is a decreased expression of our GNMT unrelated region, and also when the microRNA is silenced by an antimer. Here, one of the first experiments was performed in a bile duct ligation model of fibrosis. The expression of GNMT is restored. In this work, it was characterized as one of the main contributors of the GNMT downregulation observed during fibrosis, and when silenced, the GNMT expression was recovered. So the balance between SAMI-SACH restored prevented the DNA hypermethylation that may lead to the development of the disease. In the other work, published in 2019, I think, the role of GNMT in the mitochondria was characterized. GNMT may bound to the complex 2, so the inhibition of the microRNA may restore the expression of GNMT, so an increased beta oxidation may reduce the amount of lipid droplets, thus preventing the development of NAFLD. So in this work, as in the talk, the title of the talk says, we have characterized, Ruben and Naroa have characterized how the restoring of the ammonia metabolism protects the liver from acetaminophen. Giving a brief overview of the models assay, we have used the PAP model as it mimics the DIli observed in most of cases, which consists on giving mice, administering mice intraperitoneously 360 milligrams per kilo of acetaminophen, and then sacrifice at 2 days later. When treating the mice, we want to cover an uncovered region, because most of you already know the only treatment, one of the only treatment available is the administration of NAC, which is only effective at a certain time. So we aim to cover the time frame that is not covered by the NAC. So we chose 24 hours. And we also have performed experiments in primary hepatocytes obtained by perfusion through collagenase. We have cultured them and we have treated them with different types of acetaminophen. So, and we have collected and performed several analyses. Well, the first characterization we realized is our group previously characterized and even developed a staining method for characterizing the ammonium accumulation in the liver. As you can observe here, there is an ammonia accumulation in those mice treated with acetaminophen, and as expected, the scavenging of the ammonium by glutamine synthase was markedly decreased. It is particular that the urea cycle shows an increased activity, but as I will mention later, it may be a compensatory effect. And also, as expected, the production of acetaminophen is increased by an increased expression of glutaminase 1. It must be mentioned that the isoform 1 is the high affinity one, and is from the two isoforms is the one more related to liver pathologies. Following with this, the urea cycle is connected to polyamine synthesis, and also methionine cycle is linked to polyamine synthesis. So we perform a metabolomic characterization of several metabolites, and we observed that there was a decreased amount of the polyamines spermidine and spermine in the liver, an increase of the carboxyadenosylmethionine, S-adenosylmethionine, and also a decrease of S-adenosylmethionine. And as expected, there was a GNMT downregulation derived probably from the effect of the acetaminophen. Similarly, there was a GNMT downregulation in primary hepatocytes treated with acetaminophen, and here we started characterizing the expression levels of the microRNA. None was previously published, and here we observe that probably in relationship with the GNMT downregulation, when hepatocytes are treated with acetaminophen, there is an increased expression of the microRNA. When analyzing the liver from those mice, we can observe how those treated with acetaminophen show a higher expression of the microRNA. And we also check the levels of the microRNA in serum from patients, from DIli patients, provided by Marisabel Lucena, and we also observe an increased expression of the microRNA. It's interesting also that the increased expression of the microRNA in clinical DIli shows a correlation with some markers of liver damage, such as aspartate and alanine aminotransferase. And it's not significant, but there is a tendency, a correlation with alkaline phosphatase as an indicator of damage. So we moved to an in vitro model. We treated the primary hepatocytes with a PAP, and we performed the downregulation of the microRNA with the anti-MIR. And as you can observe here in the left side panel, when we silence the expression of the microRNA, there is a protection against the damage induced by a PAP. The relevance of the GNMT restoration when the anti-MIR is applied, GNMT is restored. But in a mouse where GNMT is not expressed, is more prone to suffer from damage, and when we silence the microRNA, we observe no restoration and no prevention of hepatocyte damage derived from a PAP. Here, similar to the in vitro findings, in those mice treated with acetaminophen and treated with the anti-immune therapy, as I have previously mentioned, we observed the downregulation of the microRNA, and related to this, the restoration of GNMT. And the effect of such restoration by the microRNA is the prevention of necrotic areas in the liver, and also the decrease in the serum transaminases as another indicator of liver damage. Also, as you can see in the left image, there is a prevention in the inflammatory response. During acetaminophen, we will have the macrophage infiltration derived from above damage, and when we treated the mice, such a staining was decreased. The TNF, although it is important in some cases for liver regeneration, here we used it as an indicator of the inflammatory response, which at 2 days after treatment was not of relevance for liver regeneration. And we can observe there is a decreased TNF expression in the TNF content in serum from those mice treated with the anti-MIR. And similar to this, the expression of several cytokines, such as CCL5, for example, was decreased when treating the mice. When moving to the in vitro model, we observed a reduction in the reactive oxygen species measured by mitosocs, which was increased upon APAP treatment and decreased when treating the hepatocyte with the anti-MIR, and also the mitotracker staining, which allows to characterize the amount of functional mitochondria, was not decreased when treating the hepatocytes with anti-MIR. And similar to this, the seahorse analysis of the primary hepatocytes show how those hepatocytes treated with anti-MIR show an increased respiration at basal levels in ATP-linked response and maximal respiration levels. We move further in order to characterize the first image was the ammonium perturbation that occurs during acetaminophen. So here we characterize the role of and the effect of the microRNA in modulating ammonium homeostasis, and we observed that it was restored. Ammonium levels in the liver were decreased upon anti-MIR treatment, and glutamine synthesis was recovered. Also, we were concerned about the possible development of secondary effects, especially the possible development of hepatic encephalopathy in DIli patients derived from the ammonium accumulation in serum. And as you can observe, there is an increased content of ammonium in the serum and a decrease by the anti-MIR therapy, suggesting that the downregulation in the liver is preventing the systemic hyperammonemia. And it is particular that when we have less ammonium levels in the liver, the urea cycle activity of the enzymes CYP1 and OTC as key enzymes from the cycle was reduced by the anti-MIR therapy. So the possible compensatory effect of an increased urea cycle activity, which as we observe is incapable of scavenging all the ammonia, is decreased when hyperammonemia is prevented. We have to remember also that here we will have the ornithine, which is from the urea cycle, and it is connected to polyamine synthesis by ornithine descarboxylase, which converts ornithine into putrescine, and then by the conjugating to the descarboxy-adenosyl methionine, it's converted to the polyamines spermidine and spermidine. They have a role in protecting the mitochondria in many conditions, and we aim to characterize if the loss of the polyamines observed when treating the mice with APAP was prevented by the microRNA. And as we can observe, some S-adenosyl methionine or ADOMET levels were restored when treating the hepatocytes with the anti-MIR, and the levels of spermidine were restored. The observed increase in the descarboxy-adenosyl methionine levels observed in those mice treated with APAP was lost, and also the decrease of spermine levels was not recovered. Further proof that there was an increased polyamine synthesis by the anti-immune therapy, we performed a fluxomics analysis. We treated the hepatocytes with labeled methionine, and by this we were able to trace and label some metabolites in the cycle. And as you can observe, there was an increased methionine levels in those hepatocytes treated with anti-immune, and an increased S-adenosyl methionine levels in those hepatocytes treated with anti-immune. On the other hand, the carboxyadenosyl methionine levels was decreased because it was probably being converted into spermidine and spermine, as the levels of those polyamines are increased in those hepatocytes treated with anti-immune, which further proves that an increased methionine cycle is fueling the polyamine synthesis in order to synthesize those polyamines and protecting the liver. To further prove this, we perform two approaches. On one hand, we have said that a higher ornithine availability may lead to an increased polyamine synthesis. So on one hand, we increase the activity of the urea cycle by providing ammonium to the cells. These concentrations is not toxic because, as you can see in the tunnel staining, at 0.5 millimolar of ammonium, hepatocytes do not show a higher death rate, but which it is observed is that in those hepatocytes treated with anti-immune, at 0 millimolar of ammonium, they are prevented from APAP-induced liver injury, but when the urea cycle is increased, such prevention does not occur. And similar happens in the other approach, where we inhibited the production of the synthesis of polyamines by those inhibitors, which prevent the protection of the anti-immune upon APAP damage. So those polyamines may be promoting liver regeneration due to their role in protecting mitochondria and also in promoting such regeneration effect. Here we can say observe the cyclin D1 and the PCNA stainings as indicators of the regeneration capacity of the liver. Upon APAP treatment, those markers are increased, probably trying to compensate the hepatocyte damage, but there is an enhanced response when treating those mice with the anti-immune. And similar to this, the relative determination of other markers and also beta-catenin as indicators of liver regeneration are increased in those mice treated with APAP. So here a brief summary has been, we have prepared a brief summary. The anti-immune therapy may restore the GNMT expression, diminishing the amount of loss and preventing the development of necrosis. And an increased methionine cycle at the same time with a recovery of ammonium homeostasis and a downregulation of the urea cycle may be enhancing the synthesis of polyamines to promote hepatocyte regeneration to inhibit and counteract the effect of the APAP during drug-induced liver injury. So regarding the other project, this project has been mainly carried out by Ruben. I would like to give a brief overview of alcohol liver damage. It is mainly produced by the accumulation of two metabolites, the acetaldehyde and the acetate, and also by the accumulation of reduction power in form of NADH, the reduced cofactor. There is an increase of response due to the metabolism of alcohol by CYP71, And the accumulation of acetaldehyd and acetate together with the accumulation of NDH leads to an increased acetylation and an increased lipid accumulation in the liver. And also to the development of the compensated CM effect, the development of inflammation that further leads to fibrosis and related complications to the pathology. Similar to what we perform in drug-induced liver injury, we checked the expression levels of the microRNA in patients, mouse, and primary hepatocytes. And as you can observe, there is an increased microRNA regulation related to a decreased GNMT expression. Also, when treating mice with the anti-MIR, we observe a similar effect which happened with DIli. I mean, the increased expression of the microRNA in those mice was decreased, and related to this, GNMT was recovered. There is a prevention in cell death observed by tunnel and the caspase-3 fluorescence activity. And also, similar to what happened in DIli, there was a decreased ROS production and also a decreased ER stress response to alcohol damage. Also, the catalyzed activity as an indicator of the clearance capacity of the liver was increased, and the amount of hepatic ethanol levels were decreased by the treatment. And similar to the effect in inflammation of the anti-microRNA therapy in DIli, the reduced glutathione levels were recovered by the anti-MIR therapy. When trying to elucidate the mechanism of action, the proteomic analysis pointed out and highlighted the effect of SIRT1, which is the acetylase that needs an NAD to acetylate the proteins. When we perform the activity of this enzyme, we observe how the anti-MIR therapy restores the levels of SIRT1 activity. And related to this, the acetylated levels in the liver were markedly decreased. And also, we were concerned because we have mentioned that during ethanol, we have an increase in NADH levels. So we are not interested in metabolizing NAD. But when we check the ratio between reduced and oxidized NAD levels, we didn't observe an increase derived from the NAD consumption by SIRT1, but we also even observe a decrease and a restoration of the reduction amount, the amount of reduced NADH levels. When trying to elucidate this effect, we observe that the relative expression of almost all the enzymes involved in NAD synthesis was increased when treating the hepatocytes with and the mice by the anti-MIR therapy. And similar to what we performed in DIli, the inhibition of SIRT1 and also the synthesis of NAD from NAMT prevented did not show the preventive effect of the anti-MIR therapy observed by when treating those hepatocytes with the inhibitors. So the conclusion of this work are mainly due to point out the effect of the anti-MIR as a potential effective therapy against the hepatotoxicity, not only for DIli, but also for alcohol-induced hepatotoxicity, because there is observed an upregulation in DIli and alcohol liver damage. And also, always, it decreases the expression of GNMT, which is of relevance for the correct functioning of the hepatocyte. The targeting of the microRNA, as expected, restores the expression of GNMT, and it protects the hepatocyte against the Synavitis, APAP, and ethanol. And in DIli, the recovery of the ammonia homeostasis reduces the urea cycle activity, which makes a higher ornithine availability, for promotion of the polyamine synthesis. And in case of alcoholic liver damage, the CYR1A activation reduces the acetylation-derived damage while rewiring the NAD synthesis. And finally, I would like to thank all the collaborators and of the teammates, especially to Ruben, Narva, and Malu for giving me the opportunity of helping in this project. And I'm here in order to willing to answer all your concerns. Thank you.
Thank you, Jorge. It was really impressive data. Are there any questions among the audience? I don't see anyone with the hands. Well, I will start asking you a few questions. One of the things that I saw from the data is that you injected 360 milligram per kilo of APAP. Mm-hmm. Why did you choose this dose? It was because it was sublethal. Why not to choose like 150 or 300 or even a more lethal dose?
Yeah, it's true that when we normally perform the experiments with the 360 dose and the 500 dose, but here we wanted to characterize if there is an increased regenerative response, which we even have characterized it and with the anti-MIR therapy. So we didn't choose the other one, the 500, because at this dose the regeneration response is not of relevance, because I mean the damage is so much that there is no regeneration. There is no possibility of regenerating.
Yeah. And when did you use the anti-MIR 873, the experiments? What time point did you choose? It was like also the same 1, 3, and 6 hours after APAP?
No, you mean in the
following experiments? Yeah.
We have to perform the transfection before, because before treating with acetaminophen, because the anti-MIR must be transfected before. Here we have a limitation because we are not seeing the treatment, we are seeing the prevention, but we don't have another choice.
And how long? So you do the treatment, and then how long after a PAP you sacrifice the mice?
You mean in the mice?
Yeah. So
no, in the mice we treat them 24 hours after a PAP administration. No. Here we see treatment because, and as I have said, we aim to cover the uncovered time frame that N-acetylcysteine does not.
N-acetylcysteine, exactly. No, because what is really striking is the effect of the anti-MIR administration on the regenerative response.
Yeah.
No? Because the cycling and the effect of regeneration. No, I think you show cycling or PCNA. I don't remember, but it's really the cell proliferation starts really. It's impressive, no? That even all the cells are positive, no? With the after the anti-MIR.
Here we have to take into account that we have an increased spermine levels that is promoting regeneration, and on the other hand, mitochondria is of relevance for liver regeneration, and by when restoring GNT and the methionine cycle and so. We observe also a higher mitochondrial function, reduced ROS, which is maybe even promoting regeneration together with polyamines.
And this effect would be also the same? I mean, it's just a hypothesis in partial hepatectomy. If you perform partial hepatectomy, you would expect so impressive regenerative response?
I think we haven't checked it yet, but it may be a good point to check it, because probably it may have an effect in the restoration of GMT. May help to further regenerate in the partial hepatectomy model.
Okay. Another question that I have is the ammonium. Is there any difference in DIli, or it has been described the difference in levels of ammonia in patients that arrive to to the emergency, and they have differences in levels of ammonium depending on the drug or on DIli damage, or is this reported, or is the first time that that you?
We only have checked it in mice, but yes, it is described because in the +1 that our group published in 2017, where we characterized and we show the method for staining ammonium, the characterization was performed in preclinical and clinical DIli.
Okay.
And we observed in both cases an upregulation of ammonia levels in the liver. And also, those patients sometimes are related to the development of hepatic encephalopathy. That's why we measure the ammonia.
Yes. Yeah, this is very, very interesting. Well, I mean, it's very, very nice. Does anyone have any questions from the audience? I don't want to be the only one asking. Let me see if I see any in the chat. If I see any, no, I don't see any questions. No questions from anyone in the audience?
Everything was clear.
Everything was crystal clear. Oh. I guess.
Thank you.
Yeah, I don't see any questions. So what is the future of the project? Do you want to? I mean, what the part that Ruben did with the with CIRTwin1, I think is really also very, very interesting, no?
Yeah.
What do you think? How do you envision the project in the future?
Well, we want to publish these results. We want to further characterize the effect of the acetylation in because we have to remember that when we have an ethanol damage, the amount of acetate leads to an increased acetylation, and we have observed a downregulation probably mediated by CIRT. So we have observed, we have performed an IP, and we have observed that FXR was acetylated by when mice were treated with the NIA model, and when were they treated with the anti-MIR, those acetylated FXR were decreased. Okay. The acetylation in FXR inhibits the protein. So with anti-MIR, the unacetylated FXR, which is the one which is not acetylated, may restore the bile acid homeostasis at the same time. Because we have observed, I don't remember because we have obtained them really recently, but the levels of some bile acids were increased by the ethanol response, probably due to all the hepatic alteration that occurred during the pathology. And when those mice are treated with anti-MIR, they show less bile acid levels. And what happens with the levels of alkaline phosphatase? Because you had them also changed with the APAP, no? What happens with alcohol in these mice? They also, when you treat them with the anti-MIR? I will have to check it because I don't remember right now. No, I mean, just because if you're changing the bile acid, I mean, the acetylation, no? And it's really interesting that after ethanol, no? This is quite novel, and it's not very investigated in detail, no? What happens, the effect of alcohol on bile acid signaling, specifically on FXR, and this is nice. Yeah, there are works that focus on the bile acid, the role of bile acids, and the gut liver axis modulation. There are some approaches that focus on this, but they are only focused on FXR and FGF15, but they are not focused on the modulation of FXR. Yeah. The anti-MIR may offer an indirect modulation of the candidate, the other target, so it may be of interest.
Yeah.
Because on one hand, we are recovering GMT, but on the other, we are restoring the homeostasis of the bile acids mediated by FXR. So,
yeah, I agree. No, it's really interesting, the project. Let's see if we have any other questions. Anyone from the audience wants to ask something? No? Okay, so then I think that's, I don't want to ask you many other questions because I have a lot, but this was, I think the most important ones, I have them very clear now. So anyway, Jorge, thank you very much. Oh, Ruben, yes, that's Ruben, the second part of your talk. No, the smallest last part of your talk.
Yeah.
Well, Jorge, thank you very much for showing us this data. And
yeah, thank you too for the invitation.
No, you're very welcome, and we hope that to keep following your data in the future and see how everything progresses very well. Okay, so the next Young seminar will be in July the 20th, and we will have Sabine Werner from Munich. And she will give us, because we had a couple of basic seminars, and the next one will be a little bit more clinic, so everyone can join, and we have a little bit of DIli and in the clinic. So until then, I hope that you have a nice month ahead. Okay, see you soon, and thank you, Jorge, again. Bye bye.